Welcome to this YouTube channel. If you are someone who is preparing for the USMLE Step 1, I want to make this journey very easy for you because it was a little hard for me, considering that I am someone who really learns through videos. So there was no really a complete playlist where I could find all of the videos related to Step 1 where I could actually focus while reading the first aid. So just to clarify, my name is not Ash. I will be revealing my name after a few videos, but let's get going. We will be starting with biochemistry, as most of you who are done with your first pass often struggle with biochemistry. So let's go. Before we start, make sure to like, subscribe and comment, because I'm making these videos. I'm taking out a lot of time to make these videos, and do leave a prayer that I pass my Step 1. If I do pass my Step 1, I will host a giveaway where I will be tutoring one student completely free of cost till they pass their Step 1. So let's go. So if you talk about the biochemistry, I had originally annotated my book, but I deleted the annotations just for you guys so we can have a fresh start. So let's start. So we know that DNA is a very long structure, and it has a lot of genes and a lot of segments. So we cannot fit the entire long length of DNA in one cell, right? And let alone its nucleus. So basically DNA is super condensed structure, and it basically what it does is that it is super coiled along the nucleosome like this. So in the nucleosome there is an octet formation. So basically octamer formation होती है जिसके अंदर it's basically coiled, and it is connected to histone proteins. So what are histone proteins? They are basically positively charged proteins that are basically large, and they have positive charge due to lysine and arginine, and it basically binds DNA to allow it to stay in a condensed form. Besides that, we need to remember that there is also an H1 histone linker that links the nucleosomes with each other. We need to remember that there are two forms of DNA. The first one is the Chromatin, which is due to histone acetylation, and then we have heterochromatin, which is due to histone deacetylation. So we are going to go over these again. So just to cover this whole part of chromatin structure, we need to remember that DNA is very large. It needs to be condensed, and nucleosome helps it to stay condensed. The nucleosome consists of the 4 histone proteins, including the H2A, H2B, H3 and H4, and there is the H1 linker DNA. We need to remember that this allows the DNA to stay in a condensed format. However, the histone acetylation converts it into an transcriptionally active form, so it can be converted into messenger RNA and then further proteins, and whereas the heterochromatin undergoes histone deacetylation, so it is transcriptionally inactive. So DNA has negative charge due to phosphate groups, whereas histones have negative charge due to I keep missing that, I'm sorry. DNA has negative charge due to phosphate groups, as we studied in chemistry that the phosphate groups have negative charge. So histones, they are large and they have positive charge due to lysine and arginine. DNA and histone synthesis occurs during the S phase, which is something I think they can ask you in questions as well. Then we have mitochondria. Mitochondria makes its own DNA, which is circular, and it does not bind histones. We need to remember this because they could ask you a question regarding which histone proteins mitochondria would bind to. So we could say that it does not bind to histones. This could be a trap question in STEP. Obviously it would not be this easy. Besides that, we have already discussed the heterochromatin and the euchromatin in a little bit, though just to revise that, we can see the lighter areas which are euchromatin and the darker areas which are heterochromatin. So these lighter areas, they are transcriptionally active, whereas the darker areas, they are transcriptionally inaccessible and inactive. So they have increased methylation and decreased acetylation. So methylation makes DNA mute, so heterochromatin basically makes it inaccessible to undergo transcription. Then we have bar bodies, which are examples. So basically they are inactive X chromosome that may be visible on the periphery of the nucleus. Then we have the euchromatin, which is less condensed and it appears lighter on electron microscopy, like I showed over here. Then we have the DNA methylation. We need to remember that the DNA methylation is involved in multiple processes that are used to alter the DNA. Just before I go on, guys, I am bilingual. I can speak both Urdu and English, so at times you might see that I am speaking in Urdu, but I will repeat everything in English as well, because I want to make my message reach a wider audience. So basically DNA methylation involved with aging, carcinogenesis. So aging and carcinogenesis are self-explanatory, that it is involved as cells age, and carcinogenesis is basically when one cell turns into cancer cell. So that is carcinogenesis. But sometimes we are confused when it comes to epigenetics, genomic imprinting, transposable element repression, and X chromosome inactivation. So just to describe that, the epigenetic changes are basically when there is no change in genetic sequence but there is change in DNA. Then we have genomic imprinting. It's implicated in disorders like Prader-Willi syndrome and Angelman syndrome, where either one of the maternal alleles or one of the father's alleles, copy of father's alleles, is silenced. So that particular healthy copy is not transmitted to the next person. Because I mess it up every time and just want to revise it, Prader-Willi is basically mother also has ER at the end, and Prader also has ER at the end. So Prader-Willi is something that is due to silencing of the maternal allele, and obviously the next one left is Angelman, which is due to paternal allele. So DNA is methylated imprinting. Methylation is within the gene promoter. So the second thing that we need to remember is that dysregulated DNA methylation is also implicated in Fragile X syndrome, which is a very high yield topic for step 1. If we talk about histone methylation, this usually causes reversible transcription expression, but also causes activation depending on the location of methyl groups. So you basically need to remember that it mostly makes DNA mute. However, there may be cases where the activation may also be caused. So we will be just going over these examples later. Then we have lysine and arginine that can be methylated, and histone acetylation and deacetylation I already discussed. So but we need to remember that acetylation basically relaxes the DNA. So when you relax, you can make proteins. You can think of it like that. So you can, when you relax, only then you can make proteins. So this is involved in thyroid hormone synthesis, may be implicated in hyperthyroidism if there is over acetylation, and in case of histone deacetylation, this is in a highly high yield topic, that is Huntington's disease. So histone deacetylation deactivates the DNA and leading to altered gene expression in Huntington disease. So that is something that we need to remember. So these are some points that we often miss while we are reading. So I think this is very important for the second pass that you go over these. But if you're watching this video for the first pass, that's fine as well, as long as you have the basic concepts. But if you have any questions regarding this particular page of biochemistry, I would be happy to answer it in the comment section. So once again, like, share and subscribe this video, and also ask your questions in the comment section. I would be happy to answer them. I would be happy to engage in any of the discussion you guys want to discuss me, and if you guys want me to make a video on a specific topic, and other than the sequence I'm going to follow for now, do let me know in the comment section so I can make the video urgently, right? Thank you so much for watching. I will see you in the next video. Goodbye.